It's right here in Conroe, Texas, and it's roller derby. Conroe Roller Derby is a nonprofit recreational league of women and men who want you to come and check out the fast-paced, hard-hitting game of roller derby. The Conroe Cutthroats practice at Rainbow Roller Rink on Tuesdays, Thursdays, and Sundays. You can find our game schedule and more information on our website, www.conroerollerderby.com.
For those of you who like your partners, your gumbo, and your music salty, well, we're here to help with the music. Julian Shea here, host of Lone Star Country Nights Thursday, your weekly dose of roots and Americana and all the music that makes this part of the country special. We stir in Western swing, honky-tonk, Zydeco, Texas blues, outlaw country, and put a pinch of red dirt, and then we smoke it over a slow fire. Then listen to the results Thursday nights on Conroe's 104.5 and 106.1 and worldwide at irlonestar.com.
Remember to download the Lone Star Community Radio app from your Google Play Apple Store. Bring Montgomery County's community radio with you anywhere with your smartphone or tablet. If you are in the Conroe area, tune in on FM 104.5, 106.1. If you are on your computer, bookmark irlonestar.com as your internet radio station. Lone Star Community Radio broadcasting 24/7 from the heart of downtown Conroe, Texas.
You're listening to Lone Star Community Radio on 104.5 KCZW LP Conroe and 106.1 KCCZ LP Conroe and worldwide at irlonestar.com.
I want to thank our sponsor, Planet Ford. Planet Ford has always been a proud supporter of law enforcement in the community, providing customer service and fleet management, sales, and service. If you're looking for that personal quality service, contact Planet Ford in Spring or online at planetford.com. Thank you for joining us on Crime Scene Today. I'm Dan Zintek, your host. Today our guest is Dr. Sibyl Bucheli, a forensic entomologist, associate professor at Sam Houston State University. Teaches contemporary biology, zoology, forensic entomology, metacriminal entomology, and evolution. Her current research studying insects as it relates to postmortem interval on human remains at the Southwestern Texas Applied Forensic Science Facility. Thank you for joining us today, Sibyl.
Hi, thanks for having me.
So, to start off, obviously I'm sure you've been asked this so many times in your career. What is a forensic entomologist?
So, to start with, an entomologist is, of course, somebody who studies insects, and a forensic entomologist is somebody who can take information about insect biology and apply it to particular situations to help answer questions about those situations. And so when people think of forensic entomology, I think that kind of Gil Grissom from CSI kind of pops into their mind, that TV show. And so they associate forensic entomology mostly with cadaver research, but it can also encompass areas such as agriculture, stored pest products, sometimes traffic accidents without identified situations are caused by people kind of freaking out over a bee or a spider being in their car, right? So it can be a really big, broad topic.
Now, we're going to get into numerous things that you've studied, your research and things, but sort of back up, how long have you wanted to do this?
It's hard to say. My mother says I was born this way. So I know I'm quoting Lady Gaga, that song of hers, but my mom says I was born this way. I have always loved insects, and so as far back as I can remember, I've always wanted to be an entomologist. And then when I was at Ohio State, I did some work with some of the entomologists there, and it just kind of grew into what it is today. Although when I was doing my dissertation work at Ohio State, my focus was not exactly the same as it is now. So I studied moths, and not really flies. So.
Okay. So in most of the forensic entomology I'm familiar with, as far as what we do on crime scenes, and my very limited knowledge of it is the insect activity as it relates to trying to come up with time of death. It's
Yes,
something
sir.
that is very hard to determine. Many times we find a body and knowing, at least in general, when they died may limit suspects. It may pinpoint suspects. And so most of the studies I've heard about have been with flies. I know that's not the only part of it, but generally, am I on the right track? That's mostly what this is, or is it much bigger than that?
I think it can take on a couple different trajectories. And so in some situations, it might just involve studying the flies. In other situations, it might encompass the entire ecosystem of insects and maybe additional arthropods. And now, and this is still far out from being used in law enforcement, although I think we're getting closer and closer. Now we're looking at how insects, bacteria, and cadavers interact, right? So it can be very focused on just maggots, or it can be really, really broad. And sometimes, I don't know, each situation is unique, which I'm sure you could attest to. But
So now, one thing that's also very unique. So now you're at, what most investigators, and we refer to as a body farm. And I know that most scientists do not like that term. And we're told many times we don't grow bodies, right? These are people that have donated their
Yes,
body to
sir.
science, and we certainly have great respect for
Absolutely.
them and great appreciation for the research that their donation has provided for us.
Yes.
But you work at you work for SAM, but this facility is part of SAM and provided up there. How many of these facilities are there? I know last count I knew the original one was in Tennessee.
Yes, sir.
Yes.
Yes.
And what since then?
So I think we're up to 6. I think so, and I'm going to have trouble listing them, but
Well, 2 are in Texas.
2 are in Texas, Tennessee, Colorado has one, I think South Carolina, and then maybe Indiana.
Okay. And I knew about the last I checked. I knew Colorado. I knew we had 2 in Texas, and of course the original in Tennessee, which finally was able to expand and get a little bit more than their 1 acre that they originally
Yes.
worked on for so
Yeah.
many years. But with that, we talked about the unique nature. Those areas, it seems from conversations, the environment and the variables of soil and just where those are located plays such a big factor that the research that you're doing, does it then limit the outcome of your research to specifically that region?
Yeah, yes and no. So we're at, it's actually the AARC now. It's been renamed. So
Okay.
staff's the Southeast Texas Applied Forensic Science Facility is now the Applied Anatomical Research Center.
You know, we're still going to call it the body farm, right?
Yes. And so we are located about 11 miles northeast of the campus in Huntsville, Texas. And we are in the heart of the Piney Woods ecoregion. And so kind of getting to your question, we think of so forensic entomology is a discipline that's based in biology, and it's got a couple different, driving theories, ecology, biodiversity, and then insect behavior. And so being located in the Piney Woods ecoregion, we have insects that are very specific to that fauna, right? So it's an area of the world that's dominated by pine forests. We have acidic soils. Huntsville's in subtropical, as is Houston. So we have a lot of rainfall, and it's a unique ecosystem. And then if you compare that to the one in San Marcos, the center in San Marcos, it's a much more arid region, right? With hardly any trees. The soil is different, the rainfall is different, the humidity is different. So in Huntsville, during the summer, our humidity is about 40% during the day, but at night it drops down. About midnight, it drops down to about 100%, right? So we have this really unique system. And so your question about it being unique, and my answer being yes and no, There are some generalities that we can make, right? The original research at the facility in Tennessee established the science, built a really good foundation, established some really good generalities, and then we can take that information and work more on that and make, and just come up with the parts of the science that are unique to the ecoregion that we're in.
Right. So one thing that Tennessee has determined is that everyone decomposes in the same stages. They will go through these stages to get from point A to point Z or whatever. So, they'll happen in the same order. So I guess really beyond that, it's sort of variable as to how long that takes and other things that would happen.
I'm smiling because this is something that we're kind of in the middle of in the research that I do. So I don't know if you're familiar with the publication Magget 2005, where Magget et al. Establishes a quantification technique to look at a cadaver by regions.
Okay.
The head and the neck, the trunk, and then the limbs. And then she establishes a method that you can score each of these areas of the body independently and then come up with a total score, and then you can say, well, that body is in state whatever or stage whatever of decomposition based on this score that I gave. And so that's a really kind of a breakthrough analysis because it recognizes that the body decomposes differentially. So in our research, we have we see all the time, I have not established in a publication, but we see all the time, the face decomposes much more quickly. Then the arms and the legs go, and then the trunk is usually the slowest to decompose. So yes, like this basis, the time frame, the big, large-scale time frame of the stages of decomposition, I think were established at the Tennessee Body Farm, and then we have kind of been looking at that, more specifically. And then in Texas, where we are in Huntsville, we have this really we have to deal with the sun.
Right, yes.
And we have to deal with the humidity. And, decomposition can move along quickly if the environment is moist and there's a lot of scavengers. But if the environment is too moist, then you're going to end up with adipocere formation, which is, the opposite of decomposition is preservation. Or if the sun is beating on your cadaver, you can end up with the opposite of decomposition in terms of dehydration and mummification, which is, of course, the opposite of decomp. It's a preservation method also.
We would expect seeing those in low humid, like Arizona has a lot of mummification versus what we see, the putrefaction here.
Yeah, and so I think it depends on what time of year we're placing our research cadavers, whether or not we will get, full decomposition or some kind of mummification. Like the sun can start mummification, or if we get some cold weather and it slows down the maggots, that can start some mummification. So it can be a little, I don't want to say it's not doing what Tennessee says it's doing, because it certainly is, but it can be a little bit,
Well, there's a lot of variables.
Variables, that's the word I'm looking for. Yeah, it
There's
can be
a lot of
highly
variables.
variable.
And I know that some of those you try to control
Yeah.
and try to regulate and such.
Yes.
One question that I have, and I know many have had this even in your field, and certainly y'all have done research on it, and that is, no matter if I am in an open field or if I am in a office building that is well built and there's a death that occurs and no one gets to the body, the flies will find it.
Yes.
Right?
Yes.
And so it's what gets the flies there. Okay?
Yes, yes.
Obviously in the field, obviously in a place that they already exist, very simple answer, right? They're already in that environment. But what about where they're not?
What about when they're not? I love that question. This is going to sound like a crazy answer to your question, right? I took my dog out last night. I come back in the house, and before I know it, my cat is in hunting position, making her little hunting noise, and, some big fly rode in on me. So we're talking about opportunistic animals. And the housefly, for example, right, Musca domestica, is found worldwide because it is really good at traveling with us, right? The larvae feed on a lot of different food sources, and everywhere humans have colonized, so has that fly. So some of your flies that you're going to find in an indoor setting are going to be able to just fly in when you open the door or ride in, or even in a house that might have some kind of opening that we don't know about, right? The insects have found it.
Right, so is it a smell? Is it a release of a chemical? Is there something that attracts them?
So there's a lot of research that looks into that, and that's kind of a, behavioral ecology, physiological question. And what I can tell you is that insects are covered in sensory organs. If you look at an insect, a picture of an insect taken at a high resolution, you're going to see hairs.
Right.
And what those hairs are actually sensory perception organs. And so a lot of these hairs are hollow, and a neuron extends to the tip of this hair. And these neurons are going to be chemosensors for airborne molecules, or they can, sense chemicals dissolved in liquids. But they're flying they're flying sensory organs. Like, they've got these wonderful antennae. They're covered in these neurons, and they do smell something.
Right.
And we're working on what that is. But we've placed cadavers for research, and as we're kind of unzipping the bag and placing the cadaver, flies are landing, and 3 seconds in. So
Right.
they're just amazing at being flies.
Yeah. Well, and
I love them. It's incredible.
so, and obviously at the research facility, they exist. They're already
Yes.
on other bodies. They're already on other things that are there, and no different than we've had in open fields and open areas and those type of things that we have. We have some insects and activities and things that already exist, so taking that into account, how do you factor that in when you're trying to figure out time of death? And I know that certain things, again, basic knowledge here of what I need to provide my entomologist, the weather that was happening in that area, and then certainly the current environment they were in. But beyond that, what would an entomologist want from a CSI on what was happening at that scene to help them sort of determine that?
Those are great questions. I would love, in an ideal world, absolutely the maggots, alive, some dead, preserved in ethanol. And then the weather, yes, absolutely. But then pictures of the scene, of the ecosystem.
Of what it
Field,
was in?
yeah, a field versus a forest. Was there a creek or a pond nearby, or was it just really dry? Is it highland versus lower elevation? And then I would again, like in a dream world, is it just a pine forest, or were there oak trees there too? Was there a lot of underbrush? Was there a lot of grass, or was the grass absent? Yeah, because again, we're grounded in a discipline of biology, and these are really important ecology determinants.
So now you want many maggots, and obviously depending on what stage they're in, they may not be on the body anymore.
Yeah.
So where would you want them collected from?
So we ask that, as a discipline, we ask that you look for quote unquote infestations, areas on the cadaver where there's a lot more maggots than other areas. So, and then from that group of maggots are gregarious, which means that they are found in large groups. They really, there's safety in numbers. From that mass, we would like the largest maggot and maybe even the smallest maggot, and then any size in between. And if they're not on the cadaver, as you said,
From what I remember, they leave the body for
They-
their change. I
yeah,
guess.
so they're going to pupate, and I can talk about the life cycle of the fly in a minute if you want. But yeah, they're going to leave the body, and they're going to go into the state of dormancy so that they can transform to become the fly. And some maggots are going to travel further away from the body than others. And so some are going to pupariate at the cadaver, some are going to leave to go somewhere safer, some are going to go down into the ground. But there's usually always some insect evidence.
Right. Well, and one thing that we've always been trained is that they're going for natural holes in the body.
Yes.
Okay. So if they're in an area that there should not be a natural hole, there possibly was an injury there beforehand.
That is correct. That is correct. So in my research, our first colonizing fly is, in summer in Texas, is going to be a fly in the genus Lucilia. And those flies are, they're really rural flies. They're not going to really be found in a city setting. We have other flies that are going to be found, in an area that's more inhabited. But these flies, Lucilia is going to fly to the nose. And almost always, that's the first place that the female arrives at the body gravid, which means full of eggs that are ready to be fertilized. And so a fly fertilizes the eggs as they pass out of the body. So she arrives gravid with all the eggs ready to go, and then she'll fly to the nose, and then she'll lay her eggs in the nose. Yeah. And then other flies are going to maybe have other sites that they prefer. And yeah, if you see, a lot of maggots maybe, in a belly button or in the side of the body, yeah,
that's not normal.
Yeah.
That's not their initial preference.
that is true. Yeah.
So now what we would love to have so many forensic entomologists available that they came to scenes, they help us, but there's not that many of y'all around to do that. So now you teach and you train, but your students that you teach, their goal when they finish is not usually always helping at crime scenes and stuff. Their end result, what are many things they go into?
So at SAM, we have, in biology in particular, we train only up to the level of the master's student. And at that point, they usually have to go somewhere else if they want a PhD. And so, students will go on to Texas A&M or other universities that offer a PhD in entomology. So some of them do go on to continue to do research in forensic entomology, fields of forensic biology. And then some of our graduate students have, graduated and gotten jobs in police departments. I don't really know that end of things, right? They're there to help analyze data, and so we have a lot of students doing a lot of different things. But I think like at SAM, I don't know, like I don't really let my master's students come to a crime scene with me.
Right.
And it's partly, it's to protect them from.
Right, it's
controversy. Yeah.
There's, I know that we've also had a doctor, by the way,
Yes.
out of your facility, and her and her master's students have come out to search
Yes.
for skeletal remains. Well,
Yes.
that's a lot different
Yes.
in searching for something that you may possibly find, and then put the right people on excavating versus putting students on the body that's going to be collecting evidence specifically for the crime.
That is correct. And so, that chain of command paperwork, I want it to all stay with me. And I use their research in that, they're important members of my team. For example, I have a student right now, Bethany, who is collecting all of the maggots. She's using rats as models, and she's collecting all of the maggots, identifying all of the maggots for summer and winter. And then once she has their identity established, she can come up with proportions of species in these maggot masses, and then we can take that and subsample from a cadaver. Just walk out to the facility, like literally with a soup ladle, scoop up a huge spoonful of maggots, take them back in the lab, identify all of them, and then look at the proportion of species in that subsample, and then see, is that representative of the population that she knows in detail, right? So my master's students are critical in my research program, and I think, helping to move the science along, yeah, but they don't really go to crime scenes.
So now, and you talk about identifying the different species. So what I'm curious about, obviously different flies, different maggots, do they, I guess, grow at different rates? In other words, is the decomposition of this is different because this different fly or this different maggot's there versus another species?
I think, again, yes and no. Nobody goes into biology because they like rules.
Right, right.
We have a joke at SAM. If you like rules, you should be a chemist. I think that if you're dealing in blowflies, the family Calliphoridae, their variation in their life cycle is going to be pretty minimal. But if you just have flesh flies, you might see something like you've raised, right? Something where flesh flies aren't found in the abundance that blowflies are found in, and therefore might be less efficient at removing soft tissues than the blowflies. So something or versus only coffin flies, which were the star insect in the Casey Anthony case, right? If you only have those tiny little flies, they're going to be there, and they can be indicative of a postmortem interval, but they're not going to consume flesh the way that the blowflies do. So I think that, again, the answer to your question is yes and no. It can make a difference, but it's got to be, at the level of
exclusion.
Exclusion of an entire family kind of thing.
So the big research is obviously to try to get to a time of death, which helps us great in criminal death. So how close can we get? I mean, obviously you're not going to walk in and say, because this fly's here 9:32 on Thursday, that's when they were dead.
And if somebody does that, you should send them back out the door, right?
Right.
Heck no. Within a day, 2 days, I think it just depends. It depends on the situation. It depends on what your time frame is, right? So we have two different models we use three different models we use in forensic entomology, but we can break them down right now into two big ones. Our first model is the maggot age model. And so the idea there is that the postmortem interval is going to correlate to the maggot age. And if you can establish how old your maggot is, then you have some pretty good idea of the postmortem interval. So that's why we ask for the largest maggot, because it's going to be the oldest maggot. And that is going to be useful only as long as your first colonizing fly maggot is still feeding on the body. So that is temperature dependent, which is seasonal dependent, right? So that can be useful anywhere from, the time of egg hatch up to the time of your first colonizer pupariating. So, a week to 2 weeks. And then after that, you have to start to look at, you can't just zoom in on one maggot, the age of that maggot. You kind of have to expand, and you have to look at the entire body as an ecosystem, and you have to look at how many adults are there, how many, the proportion of adults, what adults, how many maggots are on there. Are there, a lot of maggots, or, some greatly reduced fauna. We can look at beetles. And that is the succession model. So the succession models state that through time, as the body decomposes, it's going to go through really drastic changes. And these are changes that we mostly describe through dehydration. So up until bloat, the body is in very wet stages of decomposition. And then after bloat and purge, the body enters these drier states of decomposition. And the insects that you find active on a body in wet decomposition are really different than the insects you find active during dry decomposition. So we can piece together these big picture time frames by looking at who is present at the time of discovery. Plus, with features of the body, right? We always add in features of the body. Is the body bloated? Are large amounts of tissue removed? Things like that.
And I mean, 1 to 2 days may seem to a lot of people like a long time, but we have cases that a husband kills his wife, he leaves town, he comes back to find them deceased, and we can put it that this person was dead within that time frame prior to him leaving town, those type of things. So it's still very huge to us. Now, besides flies being on there, you talk about other insects and beetles and things. So do they all go for a feeding purpose? Is it just colonizing for it's a place to be?
So insects are driven by, I'm just going to say it because I'm a biologist, they're driven by food and they're driven by sex, right? So a lot of your flies are going to be, or a lot of your insects are going to be on that cadaver as a food resource. But once you're there already feeding, wouldn't it be convenient if your mate was there also? And so you're going to find that, a lot of insects are going to be able to complete their life cycle on a cadaver, and some insects can only complete their life cycle on that cadaver, whereas other insects can kind of come and go. So, we get the insects that tell us the most about what's going on are those insects that have to be on the cadaver for food and for mating because they can't be anywhere else, right? They're obligatory to that lifestyle, as opposed to so that would be something like, I'm thinking of Lucilia, Mexicana, and Exemia. Those are two species of blowflies that are exclusively flesh eaters. Their maggots are exclusively flesh eaters. The adults are liquid feeders, right? Adults don't consume pieces of meat. They have this drinking straw mechanism of feeding. And that's different than something like a housefly, where the maggots of a housefly can develop on, decomposing trash or feces or flesh. So that's not so, it's not so obligatory. Yeah.
Right.
Yeah.
So you have, I know at least one case that you've been called to testify on. I mean, do you get called often to testify in reference to your expertise in this?
I get called a lot. I'm laughing because in one day I had a Texas Ranger and detectives from Montgomery County at my office within 3 hours of each other, and my students.
They were concerned why they were there? That they couldn't relate it to
Dr.
the fact
Bucheli, what'd she do? Because some of them don't know that I do this.
Right.
They see, official people with guns going by, and they're just, Anyway, yeah. So a lot of times I think I get accessed a lot for background, and a lot. I'm willing to testify. A lot of times, I'm never called back, and I just think, well, maybe the case is settled, or I don't know. I don't know what happens. But
Well,
yeah, I type up official reports. I send them to whoever asks me, and sometimes I'm called back, sometimes I'm not.
Well, I know that you were in a high-profile case in Montgomery County, in the Swearingen case, who's still sitting on death row, that you were called because of the time that Ms. Trotter was in the National Forest and trying to determine, again, and that was really sort of the basis is the argument by the defense was that he couldn't have done it because he wasn't available at the time. And so trying to determine, but I think one of the biggest challenges you had to have in that was the time frame that had gone by and trying to find good information, which goes back to what we talked about earlier, and that is collection. And I know in one of the articles that you had written or were interviewed for, you made the comment that proper collection or the lack of proper collection can certainly lead to issues in the court. So where would someone go to be trained in the proper collection other than going to graduate school?
So at the AARC, Dr. Joan, by the way, holds training courses in the summer. And so you can visit the AARC website. If you search for SHSU, AARC, it will bring up the facility. And she hosts these training sessions, and they are geared to whatever the person taking them needs, right? So she can gear them to specifics that law enforcement might be looking for, or they can be really broad and kind of training educators at the high school or even college level. So I think that they can be tailored to what the student needs. But I am willing to say this on air. I am willing to come to you guys and bring my rolling suitcase of fun with me, right, and unpack the microscope and teach you guys what to do.
Yeah, how to collect and
yeah,
all that.
yeah, and talk to whoever wants to listen.
So how long have you been doing this for the research and all?
I have been at SAM doing research at the facility since 2006. The facility opened in 2009, but I started doing casework in 2006.
And obviously, as just about every PhD at a college is research driven. So
Yeah.
what research are you currently working on?
Oh, so much. So we actually, my students and I took a long time to establish the baseline of the fauna, of the insect fauna for Southeast Texas. And this was kind of goes back to your question about it being potentially specific to an area. We do say that forensic entomology is kind of geospecific. And you have to have a really good understanding of your species of insects in your area to be able to understand or, read the information that they're providing. So we spent a long time establishing the baseline diversity of carrion insects in Southeast Texas. We know the diversity of the flies that are here. We know when they're active. We know when they're not active. So for example, our most common winter fly is a fly that is the summer fly in Michigan. So, little things like that are important. And then we spent some really wonderful and interesting years looking at unusual activity. And my favorite unusual activity was the day that oak-feeding caterpillars decided to eat human flesh. And I had to convince lepidopterists, people who study moths and butterflies, that this was going on. And I would have, so when my student came to me, Natalie Lindgren came to me and said, Dr. B, the caterpillars are eating the skin flakes where the toenails used to be. And I was like, no, they're not.
Just blew her off.
Yeah, I was like, nuh-uh, they don't do that. And then she came back to me, she's like, they're doing it. And I was like, Natalie, you're nuts, go away. And then she came back to me with a video, and she's like, they're doing it on the cadaver, and at the time we had a decomposing alligator gar, and they were doing it there too. So it's that kind of thing now in the entomological field that we're kind of, is the most interesting things for us to document is like when things are not standard, right? We know what's going on.
When something's outside the box.
Mm-hmm,
right.
Outside the box. And a lot of the current publications in forensic entomology are kind of focusing on outside the box too. But my lab now, we're actually looking at this idea that as a body decomposes and dehydrates, the insects are going to colonize it in waves, right? And their time of colonization is driven by preference for the state of decomposition the cadaver's in, right? So that is a field of biology we call succession. It's predictable. We know when they're arriving or when they should arrive, and then we know when they should leave because they themselves had changed the habitat beyond their preference, and then they go to the next cadaver. But we know that bacteria are doing this. So bacteria are going to change through time on a cadaver such that certain communities of bacteria are going to be active at different stages of decomposition. And so we've actually taken a turn towards the microbiology area where we've been concentrating our efforts on collecting bacteria, establishing community structure of the bacteria, of the samples that we collect from, like what bacteria are there and when, and then running that through computer models that can tell us whether or not the community structure of the bacteria is predictive, right? So if I tell the computer, these are the bacteria I find on stages 1, 3, 5, and then I go to a unknown source swab and then tell the computer, these are the bacteria I have, it should be able to tell me, oh, it's in stage 3.
And I was looking, because that's something new as far as bacteria. Now, is the bacteria, and I guess it's coming from everywhere, but I mean, obviously from the body that's decomposing, then the insects themselves, and then I guess just what's in the environment, all three of those combined creating
Yes,
this
that's exactly
sequence.
right. Yeah, so we've established, so I work really closely with researcher Jessica Metcalf and Rob Knight, and Jessica's in Colorado, Rob Knight's in San Diego, and we work really closely with these two groups. And so we've been able to establish a lot of different things, but at SAM, we established that your body's going to pass through 3 major stages of microbial fauna, and now we need to fine tune or more deeply investigate those. But your first stage of microbial diversity is more similar to who you are when you're alive, right? So everybody is just this walking, talking ecosystem of microbes.
Right.
And then upon death, some of those bacteria are still active. Some of them die too, but some of them maintain activity. And then in the summer, the microbes will shift, the community structure shifts from being kind of human to fly, right? So there are a couple species of bacteria that are associated with flies. We only see them with flies. We never see them if there's no flies. And that kind of makes sense, right? Because that's when they're active. And then the body dries out. And as soon as that happens, the fly diversity, fly microbial diversity goes away too. And then the bacteria become more similar to the soil, which makes sense because we've got, rain or whatever, wind splashing those bacteria from the ground up onto the cadavers. So we see these 3 major stages of microbial succession. And so, we're trying to fine tune those now. So so if you ask me, I don't say, well, it's just in wet decomp right now. Maybe I could say like, oh, that's 266 accumulated degree hours. That's our goal.
Yeah, so if you break this into the stages, so the end result, we could possibly narrow down time frames. So as we said before, right now we're about maybe a day, 2 days, 3, depending on variables or whatever.
Yeah.
Where do you think this would take us?
So right now, and this is the paper. This is data from a paper that we published in Metcalf et al. In Science. Right now, we're at about 2.5 days with microbial diversity, but so it's not necessarily at this point doing better than entomological data. But what if there are no insects, right? What if, like, as time, how do I want to say this? We live in really nice houses, and these houses are really nice and sealed up. And what if the flies don't get in there, and there's no maggots to turn to? Well, maybe the microbes can tell us.
Right, so I don't think I've been on a scene yet where I haven't had a fly there.
As a forensic entomologist, I am thrilled to hear that.
They seem to find the body no matter how good the place is or not.
Yeah.
What do you see? I mean, obviously this is huge. This is a big thing. What do you see as the future of forensic entomology? I mean, where you see this going 10 years, 20 years? What do you see developing that you can't even grasp really right now? It's just sort of a thought that you're heading towards.
That's a really great question. Well, one thing that has to happen is, as I said, we're lucky here in East Texas because we have a facility that we can do all this research at, and it is a geospecific science. And so I think for every major ecosystem, there needs to be a facility to study human decomposition. And I don't know if your average community is going to be thrilled to hear that, but I think that to really understand this, we can't take what we know about decomposition in East Texas and apply that to Minnesota.
Well, and it's come a long way considering. I mean, how long was Tennessee research facility around?
Question.
Prior to us opening one in Texas, and then we had 2 in Texas, and so it seems to be growing at I won't say in any way an alarming rate, but it's certainly growing way more than it was. People are accepting it, but you're right. If you tell them that you're going to go and put dead bodies down the road from them, I mean, even both in Sam and in San Marcos, these are not anywhere near close to you're driving to get there, right?
Yes, that is right. Although they are building some really nice housing developments around our field station at Sam. Yeah, so the facility at SHSU is 9 acres within a larger 250-acre area that biology owns. It's our biology field station. So when you go out to visit us, right, we're surrounded by woods. Yeah.
Right.
Yeah. So, we're lucky that we can do that. And, I don't know, maybe it's harder for a university maybe that's located in some downtown metropolitan area to do that. But yeah, so So that would be a direction for future forensic entomology studies, just to kind of confirm these models that already have been established. But like I said, what we know at SAM, though, is that some of these insects that have always been traditionally thought of as late-stage feeders, sometimes they can come much earlier than we thought. So those coffin flies, for example, they're always thought of as being, preferring later stage decomp. Well, we get them at time of placement sometimes. So that kind of stuff has to really be investigated. And then, as I was saying, like the fun part for me are those anomalies. I think we need to look into that too. So in terms of forensic entomology, we can't just look at we can't just look for what we expect. We have to kind of have our minds open to things like flesh feeding, oak feeding caterpillars. So those anomalies are really important. But we're still missing a lot of basic information. Not all life cycles of all flies are known. And in fact, not all first colonizing flies are known. So, the first colonizing fly here, like I said, is not going to be the same as in San Marcos. Your flies that like the forest aren't leaving the forest, and the flies that like the desert aren't leaving the desert. So,
and it says a lot considering San Marcos is not that far away. I mean,
yeah,
so
exactly.
we're talking about 3 hours, 3.5 hours from the other facility. So
Yeah.
when you think about the span of 3.5 hours away from anywhere you are, that it could change that much, and we have 6 that's collecting the data for those regions, then
Yes.
a lot is missing.
Yes,
at
agree.
the moment.
Agree.
So now I'm going to tell a story. The first time that I met you, which is actually been a while back,
Yeah.
but I came to you because we had actually gone to the facility, and I came to you on a different case, just talk to you about. But we were at the facility doing burn research
Yeah.
on a body, and I have to say, you lit up because of the fact that we had it was one of the first times we'd ever had known accelerant that we went through research of burning, and obviously everything that's done at that facility is used to the fullest extent. So even though we had a small portion of research we needed to do to recreate a crime scene, you now had a known body, a known time, a known amount of accelerant, and could translate that into which insects colonize with an accelerant, how, did this affect how long it took them to get there? So, I mean, everything that's used there is used to the fullest extent.
Yes. Yeah.
So,
Yeah.
and I could tell your passion that day because I don't think I left the office before you ran out, because the first thing I asked is, are they still there? It's like, yes, they're still there, and you took off. So,
Yeah, I remember that.
so now,
Yeah.
in wrapping up a couple of, I'll say fun things, but different things. So I know so we've talked about flies and maggots. So I've heard of maggot dentistry, okay, where they have taken maggots instead of for root canals to actually put maggots in to eat the flesh and then change them out. I guess there's a purpose in changing them before they go to the next stage. You've heard of this?
Not with dentistry, but in other maggot therapy.
Basically, to take dead skin, right? So because it's cleaner than trying to, if they try to scrape it, they're taking the live with the dead.
Yes. So this works based on the idea that and you have to use a blowfly. It has to be flies in the family Calliphoridae, because other flies aren't going to do what you want them to do. And I'm going to go so far as say, like, you really want something like Lucilia, which is that's your first fly. That's the fly that can deal with just newly deceased tissue. But yeah, they stop at the living tissue.
So
yeah.
it's used in medical procedures now. I don't know how popular this is, but I've heard of this.
So and I just when I talk about this in my classes, like my favorite example is Gladiator, the beginning of Gladiator, where Russell Crowe's character, he's got that wound in his arm, and then he's in this procession to Zanzibar, and his friend is walking alongside him, and he wakes up long enough to see the maggots are in his wound, and he tries to pick them out, and his friend, whose name I can never remember, says, "Leave them, it will heal." Right. So, I mean, it goes way back, and maggots are used in medical treatment. Yeah.
So, in the class that I took, again, just a quick 40-hour just crime scene type class, we also took maggots to make maggot art.
Yes.
Okay, so you have your students doing this also?
Sometimes.
So, and to explain this, and I always have to tell people that this is a real thing.
Yes.
Okay. So we would take the maggots, we would dip them in paint and just literally throw them on a canvas. They would crawl to their death because obviously in using acrylic paint they can no longer breathe, I guess, and so therefore they end up dying because they're in a solid case of paint. But after they crawl off, it creates this pattern, and many of the detectives have those hanging up in their office
That's
now.
awesome.
And you can actually go to maggotart.com if you don't believe this, and you can see that. I think they even sell maggots online. I'm not positive why. I think you could probably make your own very simply.
Well, I know that you can buy them off of eBay.
Yeah.
Because for bait
For bait.
and or friends.
Okay. So
Because maggot art would fall, I think, into the maggot friend category.
the maggot friend
Yeah.
category. I've never. I guess I've never looked for maggot art on eBay. I don't know if that's something that sells on there.
I don't know, but you can definitely buy the maggots.
You can buy the maggots, but
yeah.
I mean,
Maybe
I guess
you-
you just leave hamburger meat out, or what they have us. It was like I'm trying to think what they had us using to grow maggots in entomology class.
Probably. Well, I like beef heart.
That was something like that.
Yeah.
Yes.
It
And,
it has integrity, so it'll hold up for a while.
So
Yeah.
if you want a science project for the summer to grow some maggots at your house, go to Michael's, get some canvas and dip them in paint and throw that on the wall, and you have a great conversation piece, right?
Exactly.
So now, as we wrap up, I certainly want to give you an opportunity. You have research going on. I know that research costs money.
Yes.
And so certainly for whatever you need, please take a moment and tell people what you do and what you have for your students and anything that you want to talk about.
Well, I do want to, I know you started the time here with thanking the donors and the families of the donors for their generous gift their generous gift to the AARC facility. And as you said, without these donations, which are absolutely priceless, we couldn't do our research. And while we have a lot of fun with what we do, we take this very seriously, and it's an honor to be able to be there and do the work. And so I want to thank anybody who has been a part of that program. And our next phase of this research project that we're moving into is actually we're building anywhere between 6 and 9 sheds out at the AARC, and then we're gonna put donations in the sheds. And then what we're gonna do is look at how the built environment, like being in an enclosure, can alter the bacteria succession and possibly the insect succession. And so that's our next step. And I have a lot of students. If anybody's listening and they have students that are interested, college-age students, we have an outstanding biology program at SAM. We have an outstanding chemistry program, and then the master's program the forensics program is a master's level program. But our students that major in biology and chemistry do research with us. I couldn't do this work without them because they're my man hours. They're the reason that we can sample cadavers for insects and bacteria every 6 hours. I can't do that. I'm in the lecture hall teaching, and so they're out there doing that for me. And so I just want to say, send your smart kids to SAM because we can't get enough of them there.
So, in the sheds you're talking about, I saw a thing online for it. They're not that expensive.
Mm-mm.
You're putting in like a window unit. It's a very basic structure. How do people send you money if they want to support your projects and things you're doing? How would they get
that
in
would be
touch with you or do
fantastic.
that?
That would be fantastic. If they wanted to do that, I would a check to Friends of Biology at Sam Houston would go into my SHSU account, and then that would be used for consumables. So we need to wear things like PPEs, personal protection equipment, gloves, booties, face masks, so that we don't get our bacteria on the cadavers. And so that donation could go to biology, and then it can say on the note like for Bucheli research.
And
Thank
we're-
you.
Of course, and we're going to put your contact information. Of course, your biography and all of the research and things you're working on is on our website at crimescenetoday.com. There'll be this broadcast will also be on there to share with others and to get that word out. And if there's anything that we can put on there to share, we'll have your contact information, and that
Awesome.
way that people get in touch with you for the research you're doing and the
Thank
great
you.
impact you're having for us in the law enforcement community. So
you.
we thank you so much for being here today and joining us and just sharing your work and your passion and your information.
Thanks for having me. It was great.
Thank you.